All right, let's dive right in. We're going deep into the microscopic world of bacteria to see something absolutely amazing. We're going to watch how they copy their entire genetic blueprint with incredible speed and accuracy, and then how they solve one of the craziest storage problems in all of biology. I mean, really think about that for a second. A single E. coli cell, which is way, way smaller than the head of a pin, has a genetic code that, if you stretched it out, would be over a millimeter long. That's 1,000 times longer than the cell itself, and somehow it copies every single one of its 4.6 million letters, almost perfectly, in about the time it takes to grab a coffee. This isn't just biology, it's a mind-blowing feat of molecular engineering. So here's our game plan for this explainer. First, we'll look at the blueprint itself. Then we'll check out the proof for how it's actually copied. After that, we'll walk through the whole construction process, the kickoff, the main event, and the finishing touches. And finally, we'll see how it all gets packed away so neatly. Okay, so before we can talk about copying something, we kind of need to know what it is we're copying, right? So let's meet the star of our show, the bacterial chromosome. You know, unlike our own cells, which have all these different linear chromosomes tucked away inside a nucleus, bacteria like E. coli keep it way simpler. It's got one main chromosome, which is just this massive, closed loop of DNA. It holds all the essential instructions for life. And this whole thing, it just kind of floats around in the cell's main compartment, the cytoplasm. No fancy packaging required, or so it seems. So the big question is, how does a cell copy a double-stranded loop of DNA? For a long time, scientists had a few different ideas, but then one super elegant experiment came along and just settled the whole debate, revealing the fundamental rule of replication. And the winning idea is called the semi-conservative model. It's actually a beautiful, simple concept. The cell doesn't just build a brand new DNA molecule from scratch. No, instead, it unwinds the original double helix and uses each of those two old strands as a perfect template to build a new partner. The result? Every new DNA molecule is half old and half new. So how did they prove this? Well, back in 1958, two scientists, Matthew Melselin and Franklin Stahl, designed this absolutely brilliant experiment. They knew DNA has a lot of nitrogen, so they decided to use it as a tag. First, they grew bacteria in a broth full of a heavy version of nitrogen. Naturally, all the new DNA the bacteria made was heavy. Then, they scooped up those bacteria and dropped them into a light nitrogen broth and just let them divide. After each generation, they'd pull out the DNA and check how heavy it was. And the result was just undeniable. After one round of cell division, the DNA wasn't heavy anymore, but it wasn't light either. It was perfectly in between. A perfect hybrid. That was the smoking gun. It meant every single new DNA molecule had one old heavy strand and one new light strand. The semi-conservative model was officially the right one. Okay, now we know what happens. Let's get to the how. You should really think of replication as a construction project on a molecular scale. And like any good project, it all has to start at a very specific spot. The cell can't just start copying anywhere it wants. It has this designated starting line called the origin of replication, or ORI for short. Think of it like a specific street address. A protein called DNA is like the construction foreman. It finds this address, latches on, and then uses a bit of energy to pry open a little bubble in the DNA double helix. As soon as that bubble is open, the rest of the crew rushes in. First up is helicase, the unzipper. It clamps on and just starts racing down the DNA, separating the two strands as it goes. Now, to keep those strands from snapping right back together, these little proteins called single-strand binding proteins, or SSBs, coat them, acting like stabilizers. And finally, an enzyme called primase comes in. It's the primer because it lays down a tiny little RNA starting block that the main builder absolutely needs to get started. So the site is prepped, the crew is in place, let's get to the main event, actually building the new DNA strands. This is where all the magic happens, at a Y-shaped spot called the replication fork. And right here, we hit the biggest challenge in DNA replication. So imagine the two strands are like a two-lane highway, but traffic can only go in one direction. The main building enzyme can only drive in one specific direction, what scientists call 5' to 3'. For one of the template strands, the leading strand, this is super easy. It just drives straight, following the unzipping fork. But the other template, the lagging strand, well, it's pointed the wrong way. So how in the world does the cell solve this one-way street problem? Take a close look here. See the master builder DNA polymerase 3? On that leading strand, it's just cruising along, no problem. But now look at that other one, the lagging strand. The enzyme has to build in the complete opposite direction that the fork is moving. How can it possibly keep up?
The solution is just brilliant. It builds the lagging strand in short, separate little pieces. These are called Okazaki fragments. Basically, primase lays down a new primer. Then DNA polymerase 3 quickly synthesizes a short chunk backwards until it bumps into the last piece it made. It's kind of like building a road by paving little sections in reverse and then stitching them all together later.
But hold on a second. If you take a twisted up phone charger cable and try to pull the two ends apart really fast, what happens? The part ahead of your hands gets all wound up into a super tight, tangled knot, right? Well, the exact same thing happens to DNA. So what's stopping the whole process from grinding to a halt in a knotted up mess? Meet the unsung hero of this whole story, an enzyme called DNA gyrase. It's a type of topoisomerase, and its job is to be a molecular stress relief expert. This enzyme works just ahead of the replication fork. It literally cuts the DNA, lets it unwind to relieve all that twisting tension, and then perfectly seals it right back up. Without this guy, replication would stop in its tracks. Okay, so back on that lagging strand, we have all these Okazaki fragments. But they're not quite done. They're still disconnected, and they've got those little temporary RNA starters. So a cleanup crew has to come through. Another enzyme, DNA polymerase A, comes in and removes all the RNA primers and fills in the gaps with the proper DNA. Then, the final player, DNA ligase, swoops in and acts like molecular glue, sealing the last little nicks to make one perfect, continuous strand. Now, remember those two replication forks? They started at the same origin point but took off in opposite directions, racing around this giant circular chromosome. Well, they're about to run right into each other. On the complete opposite side of the chromosome from the start, there are these special sequences that basically act like stop signs. They're called termination, or TER sites. A protein called TUS binds right to these spots and sets up a roadblock. It literally stops the helicase from unzipping any more DNA. The two forks come to a halt. Any last little gaps are filled in, and just like that, we have two complete intertwined circles of DNA. So we did it. We have two perfect copies of this massive bacterial chromosome. But that leads us to our last big question. How on earth does the cell store a molecule that is literally 1,000 times longer than the cell itself? The answer is this amazing packing trick called supercoiling. Think about taking a long rubber band, twisting it over and over, and then twisting that twist on top of itself. It becomes this incredibly compact, dense little ball. The cell does the exact same thing with its DNA, using enzymes like our old friend DNA gyrase to manage all these twists, folding this enormous chromosome into a tiny, organized area called the nucleoid. So let's do a quick recap of our all-star molecular team. We've got helicase, the unzipper, primase, which lays down the starters, the master builder, DNA polymerase 3, the tangle prevention specialist, DNA gyrase, and finally, DNA ligase, the one that seals the deal. It's just a perfectly coordinated molecular machine. You know, it's so easy to just dismiss bacteria as these simple little organisms. But the process we just walked through, this high-speed, high-fidelity copy and paste job, complete with its own proofreading and tangle prevention systems, it's honestly one of the most sophisticated pieces of engineering in the entire known universe. It's just a powerful reminder of the incredible complexity that's hidden inside even the simplest forms of life.